All right, welcome back. Let's get started. I know we are very close to the finish line, but I think more of a wrap-up session where we want to hear from the panelists some of their thoughts that they have learned and heard in the last two days, but also an opportunity to ask the panelists about their future perspectives and where the Human Cell Atlas can go in the future. So just before we start, my name is Shin. I'm one of the group leaders in Genome Institute of Singapore. And I would like to quickly introduce the panelists. So here to the right is Kian Kock, who's also from GIS in Singapore. And we have Laleh Haghverdi, on the far left. And we have Kevin Matthew Byrd. And we have Sophia George. And I think most of you have encountered and seen them throughout the last 2 days. But I think it's a great opportunity to, again, kind of recap some of the key messages that we've been emphasizing for the last 2 days, but also we'd love to hear how they're thinking about the future direction. Unfortunately, we won't have much time to get some questions from the audience, but maybe if panelists are okay, keep the responses relatively short, and then we can have the opportunity to maybe have audience ask some questions. Okay, so I think overall, there's absolutely no doubt that Human Cell Atlas will be, groundbreaking and huge amount of data that's going to be creating huge impact, not just in academic, but in translational front. Yet, we all recognize there's so much to do. There are multiple dimensions that we talked about, technologies to analytics to equity. And so I just want to ask quickly, maybe one or two sentences about how should HCA prioritize this expansion of Atlas, not only for version 1, but for the future version 2. So maybe Kian, you can start off by sharing where should we prioritize.
Yeah. Hello. Yeah, I think on my side, 2 fronts. One is really capacity building, because it's going to take a long time. You heard from Sanjuti about how a core took 17 years to build up. So if we really want to achieve diversification of the Atlas, we really need capacity building right now, training, outreach. And the second is really thinking about the goals this morning, so increasing diversity across all Atlases.
Stole my thunder. So increasing representation of underrepresented communities globally. And I would also add maybe consider having a predisposition network for identifying people with rare variants that predispose them to specific diseases.
I'll follow up. I spoke a lot about spatial during my session. As I speak with pathologists and clinicians about some of the opportunities that it comes with linking single cell and spatial to kind of future translation or clinical opportunities, one of the things that stands out is that there's kind of a coming field it's quite powerful in the idea of image translation. There's a wonderful paper just published last year using OCT, which is an ability to get a 3-dimensional non-invasive biopsy, translate that to H&E, and imagine that we can take that and start to leverage cell identities, make the spatial inferences, and find those patterns. And so I think there's this coming world where we haven't invited those individuals maybe as swiftly to the table. But I think as we start thinking about how we capacity build and think about spatial as well, I'd love to see more diverse voices in the room.
Yeah, I don't have much to say on that question, but yes, one thing that I can mention is just aligning this with the people's interests, right? For example, in Middle East, we talked about specific diseases. So there's a lot of cancer research. Okay, that's all nice, very good. Everyone wants to progress wherever they are. But I think if people work on something that they are more connected with, more motivated, so maybe that can push things faster.
So I hear a lot that there's capacity building, but also translation. I think this is also first time, or we're kind of inviting this idea of healthy and disease comparison. So maybe we could spend a little bit, because talking about prioritization, right? Capacity building for sure to enable data generation, but we also heard there's some challenges of collecting data and analyzing the data and making sense of the data, just using healthy samples. So I'm not saying this to make any commitments, but are there areas within the disease that could be more relevant and may have a greater impact? Any quick thoughts on this? I know this isn't one of the questions we prepared, but we just want to hear from the panelists, where are some of the areas that would have greatest impact for Human Cell Atlas and how the reference can be best utilized? Maybe we'll start from Kevin, because you have some clinical insights there.
Yeah, I think that one of the exciting opportunities is that we all know the human is a superorganism in which microbes are resident, sometimes inside of our cells at any one point. Obviously, the COVID pandemic, I think, catalyzed some really exciting research within this community about single-cell phenotypes and kind of both acute and chronic states. Thinking about this in the context of translation, again, in clinic, there's a number of really important diseases that have resident, latent associations with microbes. This includes viruses that remain latent, that are very hard to detect, Epstein-Barr virus, herpes simplex. Also even thinking about MTB, which hangs out resident often in tuberculosis and sometimes macrophages. And so for us, I think the opportunity for possibly starting to think about the question of association with microbe as a way to understand the cells that we've all characterized is kind of a meta-annotation. It's still a biological discovery-based question that still might kind of translate into have disease implications and maybe translational goals kind of on the back end.
I would add the risk, which I mentioned earlier, which is, and yes, it's linked to cancer, but we know that there are certain genes that significantly increase risk for certain types of diseases, in particular breast, ovarian, prostate cancer, colon cancer, pancreatic cancer, now skin cancer. And so there are people walking around with these mutations. Some of them get the disease, some of them don't, some of them get breast instead of ovary, and it's still a big question in the field as to what are the drivers behind why some women would get breast cancer instead of ovarian cancer, why some people get pancreatic instead of colon if a BRCA2. So there's a lot of opportunity with these high-risk clinics that are in most cancer centers or in health institutions to understand predisposition and what's going on, even from the perspective of people who do not have the mutation, because usually about 50% of people who get certain types of cancers have an epigenetic signature similar to what is in the case of the person with a germline mutation. And so there's opportunity to understand how cells move from normal to disease in that context, I think.
Maybe before you go on, I think I also hear a lot of multimodality here. I mean, I think for the greater part of Human Cell Atlas, we really concentrate a lot on the transcriptome quite a bit because technology is very much ready. Also, so in the context of expansion of Human Cell Atlas and disease introduction, any thoughts on multimodality and what some of the technologies could benefit by adding that layer into Human Cell Atlas? Maybe Hong can give some insights there.
Yeah, thanks, Jay. I think combining this with the previous question, thinking out of field a bit, what's the greatest impact HCA can have? I think the thing that I'm worried about in terms of a global setting is the next pandemic. So if I were to think not really about just a specific disease, but a class of diseases, really thinking about pairing community surveillance of single-cell technologies. And you've heard from a few of the talks, I think even last year in the general meeting, that you can mine the single-cell data to look at microbial reads or viral reads. So that's an additional modality, thinking about beyond the host and thinking more about the pathogen. So sort of opposite to one of the talks that we heard today.
Yeah. And anyone else want to add a bit more on the technology aspect? So what are some of the areas that could really benefit in addition to current transcriptome-based? And again, I'm kind of looking at Kevin because you also work at a lot of microbiome aspect.
I was really impressed with the technology lectures, especially in terms of single-cell scale. And I think one of the coming opportunities is thinking about the association of single-cell in the context of clinic. Regular blood draws kind of being part of normal practice, and again, as scalability and costs become effective, it becomes an opportunity for us to start to think about some of the these exciting questions in the context of longitudinal studies. Again, in large-scale opportunities, not just for PBMCs, but many of the immune cells that are actually non-invasively present in breast milk and urine, and saliva, nasal swabs. There's opportunities for us to think about the combination of kind of screening tools non-invasively that, again, allow us to ask basic science questions about some of the genetic diversity and other things about how it is we change over time and age, but also in terms of maybe finding things that might be prognostic or diagnostic for diseases that maybe are kind of subclinical as we tend to monitor our patients at a kind of a yearly screening.
So a lot of technology, multimodal, clinical data, metadata, which we heard a lot. But of course, we all need some computational and algorithms to do. So maybe I'll ask this question to Laleh about what do we need to do as a community to truly make the Human Cell Atlas database generative and really have impact of really getting that prediction right? Any thoughts on this?
All right, so I think one thing we have to appreciate is the scales, like how big is the space that we are talking in? Like if you have more than 20,000 genes, even if you consider 2 states on or off 1 or 2, then it is 2 to the 2,000. Like that is how big the space is, right? So, and again, so we will never be able to sample all this like huge space, right? So this is something that we have to take into account and then come up with a kind of more clever strategies where are the meaningful regions of this very big space that we will never be able to sample. And again, like back to this, the multimodal, even that, right? So like, I mean, so there is something that we have also considered that is like the notion of chaos, like in the mathematical term, that like no matter how close 2 things are, they can deviate exponentially from each other. And it is also right with multimodal measurements, for example, like in reprogramming, we see they have very low efficiency, I don't know, 6%, 10%. It means that the cells that they think they are the same, they are not the same. And now add more modalities. You will never get it. So this is something that I think we have to really appreciate in our computational methods, ways that we try to generate data. That is one thing that, yeah. And maybe another thing is explainability and interpretability that I want to emphasize. That is very important, right? So, for example, if we talk about like simple things about cell type labels, right? So it's not only the label of the cell that I need, but I need to know, at least tell me which features you looked to make that decision. That is yeah, I think these are the 2 things that I could mention that are immediate needs to be added or considered in any computational method. I mean, there are many other things, but yeah, these.
Absolutely. Very fascinating notion that more may not necessarily make it easier to understand and interpret. Of course, you're going to have conflicting tools and interpretations will make the community struggle a bit more, but maybe if there are solutions that can be devised with the community to better understand the functionality of the cell types could also be very, very important. But there's this conflicting notion that, yeah, we can expand the technology, but at the same time, we heard a lot today about equity and accessibility to these technologies and being able to provide these training and capabilities. So maybe I'll ask to Sophia first. Where would be the great way to start this? And if you had kind of unlimited resources on being able to initiate this effort, where would you start first?
That's a big question that you're asking me, Jay. Where would I start first? We would start with actually asking a priority for the place that we're doing this work. So not top down, but bottom up, with engagement. So doing a lot of buy-in because the sustainability of this, so even if you were to give a lot of money now, the sustainability is a challenge. Kodwani kind of gave us a really good explanation of how they set up their setup in Malawi, and their institutions in many places where we do our work. We're doing work in, I think, 8 countries in Africa and in the Caribbean, and many have considered establishing single-cell technologies, and then they've given up because of the access to the resources. So it's multiple things at the same time. So buy-in from the institutional leads, buy-in from the government, buy-in from actual regional funders, and even I would say the WHO. And then and then equipment, building the infrastructure that you would need to be able to do the work. And of course, everybody's talked about training already. And then let them decide what's important to study.
Absolutely.
Can I add one thing?
Please.
I think all that's correct, and I want to add one issue that we encountered in our CZI project was the technologies were not in the countries in which we were collaborating, primarily focused on Brazil and India, and there became a big debate between not us, the teams, but between our governments on IP. Because I think, as Aviv mentioned, there's an ability for us to find biomarkers in any of these studies, and if you factorialize 400 proteins or 500 plex RNA, at some point you can find maybe 2 markers that are actionable and commercializable, and then the question becomes who's going to own that, especially as it was brought to your lab. You ran it there, but they take it back home. And so setting up these agreements now and facilitating these discussions earlier will be very critical so that we can ensure that the collaborations that we all want can be maintained and sustained and even further expanded over time.
I think that's a very important notion of data sharing and open policy, and I think there are definitely members within HCA community that can help to facilitate a lot of and initiate a lot of this conversation at the higher level, and I think that's absolutely important factor that Human Cell Atlas genuinely believe in data sharing and being able to enable new science. But absolutely, I think as a countries and even very developed nations are still struggling with this notion of data sharing. Thanks for sharing that.
Can I add?
Yes.
So yes, there is data sharing and there are MTAs and all of that, but there still is a power differential, and it's a lot of it is trust, because a lot of places have been taken advantage of. And so some places we can go and no problem, and other places like it's a very, not even a toenail kind of millimeter, step by step, to show that we're not going to take and disappear. And so although an open science is sometimes not a concept that is easily digestible, because they know that they're not the ones that are equipped to be able to take advantage of the data that is being generated. Yeah. Like that is actually the major frustration. So even if it's open and they have it, they don't know how to use it. They don't have the ability to move it from basic to translational to into a product. Yeah. Not saying that they don't have the ability. If they had the ability, they would. But and so that is the thing about the open science, where even if it is open and you sign all these MTAs, there is still a differential.
Very interesting point. Even if when we talk about capacity building, we really have to think of the whole value chain of training, the data generation, but also interpretation, analysis, and productization, and all that. Is something that I think maybe slightly beyond the scope of what HCA can do, but I think as we are aware of this and trying to bring equitable open science, I think we have to keep in mind that it's not just data generation we're talking about. Yeah, great. We have about 10 minutes left, so I want to actually ask the audience if you guys have any questions, but before that, it's kind of tradition that during the closing remarks of panelists, we always ask this question about what they would do if they have $1 million prize to incentivize their research. But I thought we raised the bar a little bit, and if you have $10 million, because inflation and now these days we want to sequence more cells. So maybe I'll ask to all the panelists here, if you could create a $10 million prize to incentivize your research and kind of try to answer some of the unanswered questions in the atlas, what would it be? And maybe Kian, you can start us off.
I guess if I were to put my HCA hat on, really, I think that 10 million, I would use it for supporting the PBMC goals that we spoke about this morning, and really ensuring that half of that goes to underrepresented populations and countries, because CCI and Ancestry Networks was about twice of that, and I think this amount of money would really be able to spur and catalyze efforts globally. I think putting on a bit more of my personal hat, really thinking about public health, how can we deploy single-cell genomics to public health? And there are tons of cohort studies going on, tons of biobank samples, and then we've got the phenotypes perhaps now. So how can we use the single-cell profiles of biobank samples from 20 years ago and try and correlate it with current phenotypes? Like really thinking about prognostics and prognostics and being able to stratify populations for disease risk. So that's me. Yeah.
Great. Sophia?
Yeah, I think about sustainability and investment and monetizing whatever products that we would get out of it so that there is sustainability.
So I just want to pick on that. I fully agree, because $10 million can come and go.
Yeah,
so what are your...
So what are the things, the concrete things? Of course, like expanding the sequencing. I kind of alluded to my personal interest is looking at predisposition, so being more intentional and inviting more folks to collaborate and find these BRCA mutation carriers as a way to model risk for disease. I would, in so doing, I know a lot of us are doing whole genome sequencing, no pass, but I would say let's offer genetic testing, and I mean that's what I would do.
Yeah.
Genetic testing, so that those people that we're enrolling in our studies can actually have something, and then build capacity within those settings so that they can handle that information.
Sounds great. Kevin?
Okay, sounds like there's two answers possible. So there's the HCA answer, I'll give that one. I think that at least in my interest and some of our partners, would love to see spatial imaging hubs, kind of some key places, possibly one in Africa, one in Latin America, one in, let's say, in India or in Bangalore, where we've got a lot of interest but not a lot of options currently. So setting up these hubs and kind of creating an opportunity for them to get training and making sure that the next generation is able to take advantage of that, so that the discoveries based on AI, machine learning, biomarkers can be translated back to their own populations would be great. Selfishly, I would, I'll give a quick story. We had run the 5K Xenium on a bunch of Sjogren's patients. I have an interest in Sjogren's because it affects sites outside the oral cavity, which, if you know anything about the US, the dentistry oral divide has been quite seismic, and a lot of times insurance doesn't cover things in any of these discoveries when it comes to oral health. But this disease hits things outside of the oral cavity, including the tear ducts and the eyes. So we think we might be able to get approval from medical insurance. And so we ran the 5K on a bunch of these glands and found tofacitinib, which is a JAK statin inhibitor, which is already approved by Pfizer, which seemed to be a fantastic way to tamp down the autoimmunity. And we found out that Pfizer had an interest in this 3 years ago without a lot of data that it might be useful for Sjogren's. And so we think there are subtypes of this disease, and so I think I'd take the 10 million and I'd just try to endophenotype this disease and see if we can't make some gains for oral health that would be covered by medical insurance.
Sounds great. And I guess seed funding to generate more data to attract industry funding, that's sustainability there.
Yeah, transition to the preclinical to phase 1.
Right. Yeah. Laleh?
I passed that question. In the computational groups do not have any cost. Like we just hire students and travel.
just pay for a lot of GPUs, right? Yes. We were joking earlier we should just invest in NVIDIA for sustainability. No, but I really want to open it up to the audience. I think this has been a broad topic here, but just wanted to open it up, and maybe panelists and some of the leadership of HCA can also help to answer some of the questions that you may have. Some of the, no? Yes.
Thanks. Our interest is always to move to the next technology level. At the same time, we may also have to think about the knowledge gained from the existing technology and simplify it through a simple techniques like immunohistochemistry, where we can do the same cell type identification as we are done with the single cell. Just to give an example, in breast, there are 3 major epithelial cell types. From the single cell data, we could bring it down to 6 immunohistochemistry markers that can be used to identify the same cell type. So if we want to build the equity, we need to think about making it much more simpler than adding additional technology to make it more complicated. As we move forward, there has to be both sides to our efforts.
I fully agree. I think, I don't know if there are any comments from the panelists.
I agree.
Yeah. And I think just maybe as a side note, there are a lot of discussions also to bring the technology to clinics. You also have, you have to reduce the cost somehow so the patients are able to afford. So to what degree, how can we simplify these biomarkers could also be very important. Absolutely.
So I do think there is an opportunity to address your question on digital stains, especially training lots and lots of data and then take the H&E and find those 6 markers. There may be ways in which that a simple H&E or maybe even a bright field, if we're lucky enough, can start to visualize some of those things that make equity, because that's a cheap and fairly quick way to achieve that. I think the way we get there is lots and lots of data sets with lots and lots of data that we eventually collapse down. And so maybe that's the opportunity, is that we're hopefully going deep and wide with the ability to come back to something that's simple and effective for translation and clinical use.
Yes.
Well, I don't know who could answer that, but I am curious to know if HCA is partnering, because we know there's a lot of tools developed for computational analysis at all times. We all see this every time we come, like we have to a meeting, we see a lot of innovation in that side. But is HCA partnering in any way with wet lab developed, like for the technologists for the wet lab? Is there any partnership happening, because there is so much power within HCA? The groups are so well established. Like, is any of that happening, or can you share, or have you thought about that for lowering prices, for example?
You're talking about the commercial industry partners. Yeah. Yeah. I don't know if anyone have insights on that, or someone from the HCA.
I was mentioning that we see every time we come a lot of improvement related to computational development and analysis, but we don't see much regarding wet lab resources. And then I would like to know if HCA, because it's so powerful, there are so many labs, so many things going on, if you guys are also partnering with any commercial partners or academically to maybe try to lower prices of some of the technologies that we're using.
I'll use the mic to reply. Hang on, let me do it this way. I think you actually do see the reduction in costs that have happened. We've had a whole series of talks where cells were like at a cent and below. So, but what happens is that the moment that you can do it for less, people want to do more. And that's natural, because most of these techniques were developed in order to incentivize people to do bigger experiments. And if you think about it from an economics point of view, that's obvious, because otherwise all these companies will go out of business. That's actually how it operates. Now, if you think about another field, sequencing, that's exactly what happened there. When the prices dropped, it's not that people did the same amount of experiment for less money. They took the same amount of money and did more experiments. So that is what we were kind of oriented towards, because A, it's human nature, and it's very hard to fight human nature. But B, because it's actually a very large problem, we want to try to spread so that we can cover it. That's one of the ways in which it happens. I do think that there is more room to optimize experimental designs. I think we do too much on average of things that we've already exhausted, and not enough of things that we haven't done enough of. And for example, going after more genetically diverse individuals is one way in which we can expand our experimental designs to capture it. But we have gotten the payoff of being a very powerful community, because most of the innovations, not the sequencing ones, but the profiling ones, I mean, not the Ultima style innovation, that's the sequencing world, they're different. Most of the innovations we saw today and yesterday originated in this community, most of them directly in this community. And much of the way in which the companies, and I would invite people from 10x and Pars and Scale and so on to actually engage in this, but at least my historical recollection is a lot of what they do now is a result of the push that actually came from here, including the large-scale things for perturbations, which techniques would be useful for spatial, many, many things like that. And that's the back and forth that we get from it. But it doesn't it delivers lower prices. It definitely does. It's just that it seems that people are incapable of then actually paying less money. They just want to use the resources they have in order to do more. I'm the same, so I can't argue with that. I think that's what happens to us in practice. The reason I emphasize so much the computational side is that I think there you have a real opportunity. If in fact MUS does not have to profile the individuals after they've been challenged by LPS at 7 time points and she can do just 3, that's a real win. If in fact you can just do the H&E stain and learn something from it, and it's not that you would do 0 experiments later, but you would do something different than just more of the same, then that's a real win for us. But even then, I venture to say, given the size of biology, you would still use the same money to do more experiments. It's almost like irresistible. And that's because biology is so vast. It always feels unbounded. There's always something to do. Yeah. And I would say, I think we have here people from our many vendors, and their number is growing, and that's actually really good for this community, that there's multiple companies and lots of innovation. I think there's been a lot of good back and forth between them. I think we should challenge them to say, we want to do more. How can you help us do more? And that's also in their interest, because this is a very small community compared to the full world of biology. And because of the work that this community did in the last 8 to 10 years, everyone and their uncle, figure 1A is now a UMAP. That's not people who are in the Human Cell Atlas. They couldn't care less about the Human Cell Atlas. But if it weren't for the work that this community did, no one would be doing those experiments. Now they became like bread and butter. And you want to do that with the perturbations, and you want to do that with spatial technology A, B, C, D, and E, and you want to use different types of sequencing approaches. So yeah, if we do it together with our vendors and we have this nice virtuous cycle, they win in the end, and we win, and everyone wins.
I mean, just one thing, Patricia, there has always been also quite an active technology development community within the HCA, which is non-commercial, which is academic.
Sarah, that's exactly what I said. Methods that are being sold now.
Yeah. Yeah. And if you're interested, I mean, the Standards and Technology Working Group is also engaging kind of on that front. Yeah. So Holger is here, or Reed is here. People are here, to engage.
I think that's a great way to end this panel discussion. I think there's a really good insight, and with that, let's thank the panelists and Aviv and Sarah for their time. And I'd like to invite Gary, Holger, Shyam, and Chloe onto the stage for next session, which is on action items and next steps for HCA. So please.
Can we have oh, this is working. Okay, in case it was not clear from the get-go, This general meeting was organized around the working groups of HCA, right, sitting under the organizing committee. So I was a data ecosystem oversight representative. We have the diversity analysis working group and technology. So now we're going to summarize quickly the action items and some of the key points we've discussed at this meeting. And so representing the data ecosystem oversight, we had so much work done over the last year. I presented all the new metadata structure, all the revamp ingestion process. So what are the to-dos? Please make sure across all the bionetwork you engage with the different pillars of the ecosystem, finish submitting your individual data sets being integrated, work with the different groups to compile your tier 1 metadata, engage with the CAP team to submit your annotation metadata, and work with the bionetwork to finalize the tier 2 metadata, both in terms of the diversity recommendations as well as what you want to do in terms of your biological queries. This is our short-term goals for the next 2 years. We want to finalize version 1 Atlas and have a first iterations of the annotation and release it on the HCA portal before our 10-year anniversary. That is a very feasible goal, and it's a very exciting time. I have animation. He told me not to do animation. I should have listened. The medium goals over the next 2 years, which is required for version 2, is to engage in cell annotation effort. We're going to gather feedback from the community. So all of you are invited to give your feedback on the annotations across all 18 atlases being assembled. You don't need to dedicate your effort to only one. You're invited to contribute to all of them. And so we're asking you to please engage with your bionetwork. Please engage with the cell annotation team. You have contact information here as well. And the goal by the 10-year anniversary is to be able to release clearly where we have consensus across annotation, across all our first version of the atlas, and where there's disagreement and where we should put our efforts in the years to come. There's a QR code here that gets you to our landing page on HCA where we have detailed information about our documentation for every 3 tiers. And so it's metadata, 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 and none of this has to be done in a silo. We have an amazing team at the executive office, within the cell annotation team, within the HCA data repository and the integration team. Please reach out if you feel stuck at any point, and we look forward to seeing you in a couple of months and in the next general meeting and hearing more about the latest atlas you will have done. And the next one is Holger.
This works, yes. So I do have animations as well. Okay, we had a busy afternoon yesterday, launching a new program, the HCA Spatial Program, because we thought it is about time technology is ready, generating spatial temporal atlases in 4 dimensions. But we also realized during the discussions today that a spatial program also increases diversity because samples are more available. And in the end, we're generating many more samples, many more cells, because spatial technologies scale to larger cell numbers easily and probably in a short amount of time. We will have 10x, 100x more cells profiled spatially. So we broke out in the afternoon. Okay, there was another animation though. So I hope you can read it. If not, I can read it out for you now. So we had breakout sessions covering the 3 tracks, technology, analysis, and atlas building in health and disease. And here we identify key challenges which define in the end our homework moving forward. So from the technology side, it is still an open question which technology is the best fit for purpose. We talked about sample QC, so what sample is fit for purpose as well. And obviously, as a community, as a program, we will work around benchmarking and specifically pushing open source platforms to make spatial technologies more accessible. On the analysis side, cell segmentation is still a problem, so an unsolved problem, which then would allow us, though, to generate real single-cell atlases in space. A kind of important activity as a low-hanging fruit and maybe minimum viable product we identified is to design panels based on the atlases that we have been generating over the last years. That would allow to also reduce cost, but then it increases the technology or the analysis challenge then to integrate across the different panels. Metadata is still a topic, and obviously the integration then from single-cell atlases to spatial atlas, and eventually towards histopathology readouts. We also identified a strong opportunity to make a direct impact related to diagnostic and therapeutics, so biomarker discovery or target discovery for drugs. And as a community, as a next step, we have to prioritize organs, diseases, to really sit together and decide what is the first disease context to tackle moving forward. So I almost kept time. Over to you.
Holger, I decided for my presentation to increase diversity, I won't use animations. So here it is. Okay. I hope everyone's convinced there's strong motivation for advancing diversity, and basically equity is a fundamental value for all of us. That should be motivation enough, but there are many, many more motivations that our bodies are different in so many ways, molecular, cellular ways. Disease, pathology, risk is different across humanity. And so you cannot have one-size-fits-all clinical care, right? So those are strong biological clinical motivations in addition to the ethical motivation. And of course, there's the positive motivation also. Diversity is a scientific frontier. There's so much exciting work to be done there. And that's what I'm sure motivates a lot of us. Well, of course, to make any progress, to move the needle, we need concerted efforts of all of you. And that's what I hope you'll take away from this session. HCA has made a good beginning. There's a long way to go. Good intentions are not enough, as we've seen from the earlier talks today. So what we'll need to get there is funding, training in both directions, as Partha mentioned, all kinds of infrastructure, community engagement, as Sophia was talking about, and then a clear path to achieving benefit, and that has to include disease applications of our healthy atlases. So what are we recommending in the Diversity Task Force? All aspects of all major dimensions of human diversity and metadata spec, which I hope you will adopt. So we have one of the metadatas in metadata. And also we have set ambitious goals, quantitative goals. I hope we'll all hold ourselves accountable for meeting those goals going forward. And then starting a conversation on how to prioritize diseases, which will be many, many considerations, as I've mentioned here. Please join the HCA Genetic Diversity Network. The email is over there, and let's make this happen. Thank you.
So I felt compelled to follow the gradient that I identified in the presentation from fancy graphics, animation, and text to graphics and text to just text. So
trajectory analysis.
So yeah, just a blank slide after this. So for the analysis working group, we had 4 really great breakout sessions and a fantastic set of talks. And so just to summarize some of these, I think one big idea that comes out of what we've been talking about in this meeting is identifying computational challenges and connecting it to a community of people that are going to be working on those challenges. We have a huge strength of expertise in computational biology expertise in the community, and that's another kind of general idea that we need to think about how to connect the expertise to the people that want to develop expertise. So there's a lot of demand for training and learning, and we have to figure out interesting ways or good ways to make those connections. There's also an interesting spectrum of activities from cutting-edge innovative research to practical applications, and we need to figure out how to structure our activities to support that spectrum. So there are lots of interesting challenges that come up that were identified in the spatial session. Disease comparisons became more emphasized at this meeting. Figuring out how to integrate human diversity as a first-class aspect of all the analysis methods, all the way down to the AI cell, and if Dana's listening, not losing the rare cells when we integrate data. There's nice sections out, initiatives like Open Problems that can help us keep track of these challenges and advertise them. So we need to engage busy experts at the same time as bringing in a wide, diverse, and global community via different programming that includes training and working groups to engage in two-way communication around all of the different activities that we have. And that connects to the accessible tools ecosystem that we're building, so there needs to be a communication there as well. Is that it? Okay. Is that it?
Take this one.
So let's go with this one. So, okay, and all the good things will eventually come to an end, and I'd like to thank all of you for having a fantastic meeting. Thank you for coming, for participating, making this meeting very lively. Some numbers that I would like to give you. I think it was very successful, but this is a doesn't the okay, perfect. So we have so. For 543 countries is too many, I guess. There are not so many countries in the world, but anyway.
Some of us actually come from multiple countries.
Right.
Like multinationals, so maybe that's...
Right, okay. That is...
I suspect that it's
okay,
either
probably.
3 or 4 things.
Wonderful. Anyway, so we have 296 attendance in person and 161 virtual, which is makes more than 450. Many posters, 50 speakers, talks, breakout sessions. So I think that this was really great. So also we would like to hear from you. So please scan the barcode and scan the barcode and give any feedback that you would like to leave us. Recordings are available, and if you want to hear again our presentation, please go to this website, and which will contain all the presentations and the discussion that we had in this room. So, and please do not hesitate to really go there and revisit what we've been discussing. So many thanks to many people and companies. First of all, I would like to thank 10x Genomics for their foundational level support for the 2024 Human Cell Atlas General Meeting. Thank you. Without a sponsor, we cannot have those meetings. And also, I would like to thank the Helmsick Charitable Trust for their foundational level support for the 2024 Human Cell Atlas General Meetings. And also our bronze level sponsor, BioLegend, BioScurb, CIFAR, Curio, Euroclone, Scale Bioscience, and VisGen. Thank you again for them. And again, our exhibitors, Element Bioscience, Ultima Genomics, 10x Genomics, Illumina, VisGen, Takara, Scale Bioscience, Oxford Nanopore Technologies, and Pers Bioscience. And again, thank you to all of them. The program committee with Gary and Muz, Shyam, John, Aviv, Lucia, Sarah, and Alexandra-Chloe. Thank you very much for the contribution and for the fantastic leadership in all of this. We have received a lot of help from many of you, and of course we have the executive officer, Aya, and the many people from Human Technopole, and I think that they are mixed here. Anyway, those that have been running with the microphone. Thank you. Yes, make it bigger because otherwise. Aya Roberto, Aya Al Rauni, Roberto Maria, Yoshi Ando, Pietro De Mela, Altea Giorgiai, Manuel Lessi, Azahara Maria, Fuentes Trillo, Gaia Matassa, Kate Mullerin, Matisse Ozols, John Randall, Lucia Robson, Jenny Rod, Marta Salese, Christine Schwenk, Ellen Todres, and Sam Wynn. Thank you again. And also at Human Technopole, Chiara Mantovani, Bianca Montini. Thank you to all of you for being here and for the great support. And this is the picture of the executive officer. I will transfer now for the upcoming events to Sarah and Aviv.
Thank you so much, Piero. So we have the exciting announcements about events coming up. On the website, we have the HCA Asia meeting, and that's very soon in December in Hong Kong. And that will be an exciting meeting because it will be just after the Human Cell Atlas publication bundle announcement. So there's going to be an exciting winter time coming up for HCA. In your diaries, please also note 2025. So the next installation of this meeting will be 9th to 11th of June 2025, and we'll all be flying to Singapore. Woohoo! Yeah, and we have Jay and Shyam here, kind of the local delegation. We want to emphasize that anyone can contribute to the Human Cell Atlas. So please register and sign up here. And you can join the biological networks, visit the HCA website. And I will hand over to you.
I will give you some additional things that you can do. For example, you can pay attention to the key takeaways from the meeting. Okay, the slides here are like totally crazy right now. First of all, I think it's really important to engage with the data ecosystem. We need the metadata. And remember, there's 3 tiers, tier 1, tier 2, and tier 3. And if in doubt, you can always ask Chloe. We really are moving to the spatial era. It's great to profile individual cells. It's even better if we know where they are located. And so there's a lot that we need to do there. We need to do things different experimentally. We need to source the right samples. We need new analysis algorithms. We'll need the data to be processed, analyzed, and some foundational models might need to be learned. There's a lot that will need to be done, but there's a lot of upside. We will know where the cells are, and it is easier to do in a more diverse and global fashion. That brings me to our commitment to expand the diversity and advance the equity in our atlas. Funding, we will try to make it cheaper. I'm just predicting that we will then just profile more cells. We want to make sure that we are committed to community engagement. As you've heard, people need help with training, for example. It means occasionally people have to travel to places to do that training. We need to build the right infrastructure and make sure that the benefit is clear. And this is where Shyam highlighted to you how we're going to start thinking about which diseases are the right diseases to study under the premise of the Human Cell Atlas. And finally, we really need to integrate all of this information into a view that is multi-scale for the actual human body. And that means the single-cell data, the spatial data, the histopathological data, and actually it doesn't stop there. In the end, you have the entire organ and its structure and how it's all connected together. And we saw it because the brain leads the way. You can see that they're already moving in that direction. And finally, we have great opportunities for synergy. We talked a lot about our vendors. We talked a little bit about the fact that we actually want to make an impact on human disease, and so we need to engage with all parts of the ecosystem because we can only achieve that together. And in the meantime, I will wish everyone a great rest of the evening, a wonderful day tomorrow when we have some additional satellite activities. I'll get there. And really look forward to seeing everyone in Singapore 2025. And for this, I will turn to Jay and Shyam to tell you all about that. And by the way, I've never been to Singapore, so this will be my first time. There we go. I think you need, do you need this or yours? Yeah, it's not clear which ones were.
Well, we really look forward to welcoming you to Singapore, but my role here for now is I'll invite you to also HCA Asia meeting in Hong Kong. As Sarah mentioned, again, we are pushing a lot on regional science to not only equity, but also opportunity to create new exciting projects. And you probably have heard of IDA for many times. It's been a flagship project for HCA Asia to initiate and bring together the whole community. And it's been extremely successful, and we hope to build IDA 2, maybe different tissues, thinking about skin or other interesting organs that we can tackle as a regional science and regional HCA. So if you're interested or want to learn how regional network of HCA can be built, please, I invite you to come to Hong Kong in December. And for the general meeting, I'll invite Shyam to talk about Singapore.
Thanks, Jay. Excited to.
No, you need the other one.
Okay. Excited to announce the next general meeting, June 9th to 11th in Singapore. Hope you'll all come there and participate. We'll have to keep up with the energy of the amazing meeting that Piero and team have organized. We'll try to live up to the high bar. And as you saw, lots of new directions here that we discussed today. The data ecosystem that Chloe was talking about, the spatial forum that Holger introduced, the diversity initiative that just rolled out its task force recommendations, the disease applications that Aviv mentioned and many other people mentioned, and Sarah and others. These are all of them underpinned by data analytics and algorithm development. So a lot of new balloons being launched in the HCA, and hopefully when you come to Singapore, you'll see those things becoming, going further, higher up in the air, becoming more real and broader scope, and be a part of turning all those initiatives into concrete projects.
Singapore has amazing food. So it's a lot of fun, so come. Yeah.
Come for the food, stay for the science.
It is the competition Milano-Singapore, right? Okay.
All right, thank you.
One more minute before everyone leaves. There's one person we haven't thanked individually, and that is to Piero, who really did a fantastic job.
Thank you for coming.